I'm Kris Burkewitz, assistant professor in cell and developmental biology here at Vanderbilt. We're interested broadly in the biology of aging, and we tend to break that into three bigger, broader types of questions. The first being, what are the cell and molecular level changes that are occurring as we transition from youth into older age? The second, and a little more difficult, is to identify which of those molecular changes are actually linked or causing the sort of negative consequences that we think of as we think of aging, things like chronic disease risk, neurodegeneration, diabetes, and just the morbidity and frailty that accompanies old age. And the third and most difficult question, I think, is to ask whether there are ways in which we can intervene in that process to prevent or delay those changes from occurring in such a way that we can reduce or prevent the risk of chronic disease in older age. The really exciting thing about the aging field right now is that there really is proof of principle that we can achieve that goal of delaying or ameliorating the aging process. And so there are a few different strategies for doing that. And the one that we're most interested in as a lab is, or involves nutrient sensing, nutrient levels, and metabolism. There's this very ancient and deep evolutionary link between nutrient levels and the aging process. And the idea behind that is that when animals encounter the risk of starvation or starvation environments, that they can kind of flip some molecular switches and reprogram their physiology in a way that gets them through the tough times, the lean times, and allows them to make it to the conditions of food and plenty again. And one of the side effects of that physiological reprogramming is that those animals can actually live longer and age more robustly later in life. And so we can mimic some of those effects by imposing strict dietary regimens on our animal models. But to make this sort of strategy applicable to more human populations, we really need to find or understand better how those molecular switches really work so that we can develop drugs one day to mimic these sorts of benefits on the aging process. So the sort of foundational model that we use in the lab is this nematode worm C. elegans, and we use it because it has three really important experimental advantages for studying the biology of aging. The first one is that whereas certain types of experiments might take decades in primate models, years in mice, we can actually age worms out, these nematode worms out, in 3 to 4 weeks, which really allows us to run more experiments and ask bigger, riskier questions in this model. The other important element to C. elegans is that it's very easy to genetically manipulate these animals. So we can really use genetic tools to modify the nutrient sensing pathways that we're interested in, and also to modify these different cell biological events that we're following as the animals age. And finally, the last advantage is that these animals are transparent. And so that means that we can actually watch the aging process in any cell that we want in these animals at the molecular level, in living animals too, all the way from birth to death. And that's really unique among laboratory models, so we use that a lot. We are right now hypothesizing that there's a whole new realm of pathways that could be targeted by drugs one day to improve the aging process. And so we're using genetic techniques right now to ask whether the machineries that are regulating organelle structure and maintaining organelle dynamics and their healthy youthful behaviors, if we can support those types of machineries in the cell later into life, can we then maintain the metabolic function at the subcellular level and build upwards to maintain physiological health outwards from there. So I think I knew I was going to be a scientist from a very young age, so the question for me was really, what type of scientist am I going to become? And I think it really came down to just picking up a book as a high school student even about the biology of aging, and just having it suddenly hit me that aging isn't a sort of fact of life, that if I'm going to be a scientist, I need to think about what does aging really mean. And it turns out, even back then, we knew that there are certain molecular events that are common to the aging process for many different types of animals. And on top of that, there are different types of cell defense and stress pathways that can prevent or repair those types of damage that occur with aging. And so suddenly, the biology of aging became not written in stone to me, but an actual curious sort of byproduct of evolution that we can actually target in certain ways. And I think that opened up the door to a scientific problem for me that I think will take a whole career to sort of dive into the complexity of the aging process in that sense. So basic science is really everything in the aging field. I think for most of human history, aging and aging science and the attempt to extend lifespan has been the realm of snake oil salesmen to some extent and pseudoscience. And so it was really basic science efforts that began, I would say, in the last century, but that have really accelerated over the past 20 years and grown dramatically, that have really set the foundation to take aging biology from pseudoscience and setting a real foundation that we can build sort of a translational outcome on top of. So funding really enables everything that we do. We have a mixture right now of federal funding, public funding from the National Institute on Aging, as well as some small private grants. Of course, we couldn't function without what we have now, but if we can secure more funding from any source in the future, that can allow us to do things like expand our model system, expand our discoveries beyond C. elegans into vertebrates and mammals, and one day open the door for translating these things into the clinic. So I left my postdoc looking for a home for my new lab with a background in aging and nutrient signaling and molecular genetics. And the cell biology that we were interested in working on was still at that time relatively new to me. And so I identified at that point that what was going to be most important was to find a collaborative community of people that could really help us grow in those directions where we weren't already very strong. And of course, here at Vanderbilt, the Cell and Developmental Biology Department, we were elite in those respects, and so it was a no-brainer, I think, to join this group of people. On top of that, I know that the trainees here, the students, are excellent, they're top-notch, and they're well-supported. So we have great people to do the experiments, to do the research, to ask these types of questions. And I think the final reason is really that one of our main tools is microscopy. And so the resources here at Vanderbilt, the Nikon Center of Excellence, the CSER cell imaging resource here are again, elite, exceptional resources that are really enabling us to image the aging process, to observe the aging process in these models in ways that other people haven't done before. So I think that my advice for students that want to join the aging field, there's two parts of it. Part can be generalized to anyone that wants to do biomedical science, and that is to make sure that you have identified a problem, a question that you are interested in and passionate about that is deep and complex, so that the mounds of negative results that we all experience in science won't ever dissuade you from continuing in that path. More specifically in aging research, I think I would suggest that students really try to find an environment, by which I mean a mentor and an experimental system that allows you to be really creative, that allows you to take risks with your research and to ask questions that are innovative and new. I think the aging field really benefits from that type of perspective and that type of research. And so that's really all about choosing the right place to do the work that you want to do.